Kia ora, everyone, and so welcome to the next of these mini online lectures. What I'm going to do today is take our understanding of phylogeny, and in particular the terms monophyly and paraphyly that we learned from the last mini lecture, and I'm going to apply those to a problem or an issue that you've covered already with Mars, which is the idea of one fungus, one name, and the difference between sexual and asexual fungi. So let me turn my video off and we'll start going through the slides. Okay, so with Mars, you should have learned that sexual and asexual species are often given completely different names, because if we find a fungus with asexual structures, we find another fungus with asexual structures, how do we know if those two fungi are the same or different? And so one of the ways that we can do that is use DNA sequences, because DNA sequences don't care whether a fungus has a sexual structure or an asexual structure or no structure at all. And so you can use DNA sequences to resolve the correct evolutionary relationships in phylogenies, so you can get your taxonomy correct. So we're going to look at a couple of examples of that, and in the terms of phylogeny. So let's go to slide number 2. And here I'm showing an example. So here we have two genera, Calanectria and Cylindrocladum, and we've taken DNA sequences from one particular small part of the genome, so one part of a gene, and we've used those DNA sequences to construct a phylogeny. And what this phylogeny shows is the relationships between all these different species. So there's multiple different species here from these two genera. And any time that you see a vertical line like this, that means that the two individuals in question are identical to each other. So in this case, the DNA sequences for these two species are the same for that small piece of gene that we're analyzing in this thing, not for their whole genome necessarily. And so what you should be able to see is, and we'll go to this, and so Calanectria are the telomorphs, so these are the sexual species, and Cylindrocladium are the asexual anamorphic species. And so what you should be able to see is that these two genus names, Calanectria and Cylindrocladium, are both paraphyletic names. So in neither case does every member of that genus group within a clade in the phylogeny that is separate to any other genus. And in fact, what we can see is that for each species from one genus, there's an identical species from the other genus. So this tells us that that these are just telomorphs, and anamorphs of the same species. So the names of paraphyletic and taxonomically, what these should all be called with just the same genus. So these things should all be one single genus, and then the two, in each case for each of these pairs, these should have just one single species name. Okay, so hopefully that is clear. Let's go now to slide number 3. And I've shown this picture before, and so these are the class 1 endophytes, the epichloroid endophytes, and I showed this tree of the endophytes before. But now I want to just point out something that I didn't really point out in one of the previous mini lectures where I showed this. And so as I told you, for these class 1 endophytes, there's some species that are known to be sexual, and they choke they suppress reproduction of the host grass flower. And some are completely asexual, they only ever vertically transmitted. And the sexual species were called Epichloi, were put in the genus Epichloi, and the asexual species were all put in the genus Neotyphodium. And so you can see those on the tree by indicated with the genus name starting with an E and starting with an N, obviously. And so what you can see is that the Epichloi species are completely interspersed with the Neotyphodium species. So therefore, both the Epichloi genus name and the Neotyphodium genus name are paraphyletic terms. So each one doesn't create a nice monophyletic clade to the exclusion of all other or of the other genus name. And so if you were in charge of taxonomy of the Epichloi and Neotyphodium endophytes, what would your solution be to this problem? And with any luck, what you would have come up with is that you would give them all just a single genus name. And so you could call them all Epichloi, or you could call them all Neotyphodium. It happens that these have all been renamed as Epichloi, and so that's why we refer to them as the Epichloi species, but they could have equally been renamed all as Neotyphodium. But obviously they're just a single genus, and then that is therefore they should all have the same name. So that's all I want to talk about in terms of this small practical application of the use of phylogeny and the ideas of paraphyly and monophyly. What I'm going to do when I come back is we're going to take this to another level, and we're going to start to look at what we expect to see in terms of population structures and phylogenetic relationships of pathogens depending on the nature of the pathogen outbreak. But for now, I'll finish there, and if you have any questions, please address them to Piazza.